Hello, today C&T is visiting the headquarters of Sederma in France, and I'm with the marketing director Olga Grazioso, who is going to tell us a bit about the company and the latest activities and new products before we go to visit the laboratories and see the latest in technologies that they're using in product development.
Thank you very much, Jen. I'm really happy to welcome you here in our facilities, and you will see where was discovered and created our latest launch, Ambostem. This product was developed in the plant cell culture laboratory by the team of Frédéric Debin, our lab manager that you are going to visit, and this product has been conceived to take care of olive tone skin. Our in vitro laboratory, led by Thibaut Marchand, that you are going to visit as well, came up with some very interesting benefits for this product, especially fighting against redness, dullness, and bringing a bright full complexion. So this product is a co-design. It sits with our committed beauty vision that includes diversity and environmental protection and is totally in line with our sustainability engagement.
I'm Frédéric Debin from Sederma. I'm in charge of biotech department, so gathering, fermentation, so microbial culture, plant cell culture, and a part of extraction at laboratory scale. Here we are in the fermentation part, so I will explain you how we work with microorganisms to develop a process, eco-design process, to produce cosmetic active ingredients. So we work with all type of microorganisms. We can work on yeast, bacteria, microalgae, fungi, and we work closely. So we work closely with the biodiversity, and we have to take care of different things with the biodiversity, especially, for example, Nagoya Protocol, regulatory aspects. So, and we can work with every type of origin of microorganism. So we can work with water microorganism. For example, we have some products from bacteria that live in deep sea, in very constrained environment, and this is that for us that is interesting, is to collect some microorganisms that have specific properties to produce specific molecules with biological activities in terms of cosmetic ingredients. The first step is to do the research on these microorganisms, because we collect microorganisms in the nature, so we have to adapt the microorganisms in laboratory conditions. So we work on different experiments, different design of experiments by adapting the condition to the microorganisms. So for example, we can work on the culture medium and different conditions as temperature, pH, and so on. The next step is to amplify the biomass content. So we work with bioreactors to have more and more biomass to work. And why we want to have this biomass why we want to have more and more biomass is to work on what we call the chemical mapping. Chemical mapping is to know what we have in our biomass in order to select the active molecules. We have some target molecules, but we want to know also the global composition of the biomass to work specifically on the optimization of molecule production. How we can do that? We do that in the bioreactors by playing on different parameters and by playing also on the biomass. So we don't use GMO organisms. We work on the culture condition and on the process parameters to optimize our biomass. Once we have defined our optimized biomass, we work on the skin efficacy. So we want to validate by screening different biological activities. We want to know what are the properties of our future ingredient in terms of skin bioactivities. If the performances are good, in terms of skin bioactivities, we continue with the scale-up of the biomass and the scale-up of the process. If not, we come back to research activities or we can stop the project. The scale-up is done in bigger bioreactors, so we start with 2-liter bioreactors in the laboratory, but also we can go to 20 liters, 600 liters, and if we need more in terms of production, for example, we can go to 20 cubic meters with the collaboration we have in the group. In parallel of biomass scale-up, we have the downstream processing definition and optimization in terms of process. So downstream processing is how we can recover the biomass and how we can recover all the molecules of interest we have. So we can work with different technologies of clarification, filtration, also concentration with centrifugation, for example, or some other techniques of filtration. We can also do the lysis of the cells if the molecules are intracellular, or we can recover the molecules in the supernatant, in the culture medium, in fact. We can also do the purification of the molecules. In fact, we can do what we want in terms of downstream processing to recover the good molecules of interest and to have finally an active ingredient that is ready to be sold, to be industrialized. So we have, during this process, we have checked different things, marketing aspects, regulatory aspects, IP aspects also, and we are ready to produce our active ingredient. We have here some plantlets. So these plantlets have been generated from a plant. So this is our starting material also to generate and to induce calogenesis, so to induce callus production, to have the future biomass to be worked under research phase. So here you can see callus that were generated and induced from plants. So this is important to know that we can keep this aspect of cells during years and years. So it's not necessary to collect again the plant in the nature. We have our stock material, and we work with this material during all the research and development phase. And also we keep these cells during all the life of the product. We have a collection of different plants under this stage, and also we have a collection of callus. Thanks to the IRB integration in 2012, we have now probably one of the biggest collection of plant cell lines for the cosmetic industry. We have approximately 100 different cell lines that are worked and that have generate different projects and future products.